Hello. How are you?
Sort of okay. Yeah.
Is that just with your graduate program?
Yeah, just with the graduate program. They're being fussy or something, or they didn't like what I did. How do they know what I did? I haven't handed anything in, so I decided to write it up.
Oh.
So I have 5,000 words for them right now.
Okay.
And another couple thousand words. Maybe I can hand them a 10,000 word essay and say, this is what I've done. Here's 10,000 words. Do you want more?
The whole paper, yeah.
Yeah, it's 5,000 words. And it's definitely not finished.
Oh, yeah. I sent you an email with those papers. I don't know if those are the right ones.
Well, one was, and the other one looks interesting, so
okay.
It's good. I was just trying to get at how things tiled together, and
right. Good luck with that. I don't know. Usually,
it's just annoying. Like, you're gonna put more pressure on me. Like, I can't. Like,
yeah.
I can't work any faster than what I'm doing. Like,
it's not. You can't just pop it out like in a couple days.
No, and the other thing is I'm good at writing. So if you want output, best way for me to do that is to write. And like I said, 10,000 words. You want to read it, there it is. So you go ahead.
Yeah.
Sort of revenge of the nerds. Let's write this up.
That's good. Hello, Jesse. Hello, Dick. How are you guys doing?
Okay.
Good.
The students from India going to join you?
Yeah, Sushmanth was going to join. He's going to talk about something called Hacktoberfest, which we've done in the past, but it's something that involves the GitHub repositories and things like that. I don't know if he's going to be here or if he's not going to be here. He sent me Jesse said he'll mostly be listening. Just because Sushmanth sent me a message just recently. Yeah. So I'll talk about it, and I don't know. Maybe he'll come in. So yeah. So one of the things we'd like to do in the next, imminently, I guess, because it's almost October, is to host something called Hacktoberfest. So Hacktoberfest is a time that is sort of an official period for GitHub where they people expect to be able to contribute to GitHub repositories. And some organizations offer, if you have 5 commits during that month or 5 pull requests, that you get some sort of recognition or free swag, which is like a t-shirt or something like that. Oh, there he is. So I don't know if we don't have the bandwidth for swag. I don't think.
I'll shut my
okay.
camera.
But it'll be a nice thing to participate in. Especially, we've done this before. We did this, I think, 2 years ago, or maybe even last year. I can't remember. And we had a pretty good response. So people will come by the repository. In this case, it's the GitHub organization. Look it over. We have to create some issues for it first, and then people will. We can advertise those issues, and then in the meeting, for example, and we can have people work on them, and they can get some sort of recognition for it. So Sushmanth, hello.
Hi Bradley.
Hi, how are you? How are you?
Fine. Fine. You're discussing about Hacktoberfest, right?
Yeah, that's what we're talking about. I was just setting it up for you.
Okay. This is the project I was thinking. So, I have kept a YouTube video, which has a project actually. It is of fine-tuning the Detectron 2 model. But before fine-tuning, we need the data set. I was thinking to create a custom data set for us. I already took the TIFF files from the cell tracking challenge dataset and converted them into images. Right now, our plan would be creating a custom dataset. I mean, everyone need to learn the cell lineage analysis according to position. They need to name the each cell using a tool called vmakesense.io. If you can play this video, can I share my screen? And
yes, you can.
Could you see?
Not yet. I'm not sure why it's not letting you share.
Just a minute, I will rejoin. I think.
Okay, yeah. Or why don't we let him rejoin, see if he can log in and share his screen. Okay.
Hello?
Hi, how are you? Here we go. He just said don't allow.
Yeah. In this video, actually, this is the custom data set he was making using a open source library called vmakesense.ai. It is like free and open source.
Oh.
Can't you see the screen?
Yes, yes.
No? Vmakesense.ai. Yeah, this is the website I'm talking about. We can upload all our images right now what we have and make them into a object detection kind of method. If you see in this video,
yeah, I can see it.
These guys are making like a ground truth of it. So we are working with CL games, right? We will use this tool and we will annotate each name for lineage analysis. Like it will be custom object detection plus lineage analysis model. At the end, if we fine tune this model on the dataset whatever we are making for Hacktoberfest for next GSOC, it could be a combined model. It will segment the cell also, and we can get the positions of each cell, and we can also get the what we call the lineage analysis in by a single model. Actually, we have 3 different models for each thing. So by using this, we can directly segment the cells. But in Hacktoberfest point of view, we'll just make the data set because right now we have around 5,000 images of CL GAMs. We need custom data. So I was thinking to give whoever participating in Hacktoberfest, give them this data set and try to annotate them. That will be the Hacktoberfest activity. After annotating, we could extend this project if we can complete this data set on time in Hacktoberfest. They can start working on the model also, or it could be next GSOC project. Finally, it's an object detection, segmentation, and naming also. Bradley, before you taking any decision, please give a look on this video. This can be not only used for C elegans, but also in other kind of organisms.
Yeah. So this
could be some kind of yeah. Sorry.
Oh no, finish up. I was gonna say something, but
This is going to be like a first we need to make the data set before fine tuning this model. So
right.
yeah, it could be perfect. It's a large, well-trained model, so it could segment the cells properly. He just tried to fine-tune this model for this video for only for 15 or 25 images, and it is giving best results at the end. So if we make our custom data set of 5,000 images, and if we could fine-tune it, it would be act as the best model for C. elegans. I think so.
Yeah, that would be great. So you said you have a data set in mind, or
Yes, I have the data set actually. C. elegans cell tracking challenge data set.
okay.
Maybe I will provide you the data set images. I have converted all the TIFF files into images. They are grayscale images. This is how they look like. I will show you that. Yes, data. Yes. Yes. If you see Bradley, you can see them.
Yeah,
We
I
can
see.
name them using the tool. We can just use for object detection. Actually, he also have the microscopy images also when he was annotating it. So
All right.
they are a bit clear, but ours are not clear. But we can just name them. At least create a boundary for it by using this tool and name them each of cell.
All right.
Not these cells, but which are less densy. Some cells are overlapping over this thing. So go for 54. Yeah. Here they are not colliding actually. See.
Yeah, some are harder to. So this is about like
Okay.
basically having to annotate these, or with some data set.
Yeah, annotating it and creating the custom CSV files about the location and the cell name.
Okay.
Before, please give a look on this video. I hope that would be helpful for you also.
All
I
right.
have shared the video link over here.
Yeah.
Then we can decide we can continue on.
And so, if people are going to do this, what do we need to provide them to? Like, do we need to
Actually,
have?
we have nothing. Whatever software we are using is open source. MakeSense.ai. We need to divide the work actually. Whoever is participating, and give them the data set, and whoever works and completes, we can issue them swacks. Maybe we can give a 50 images to each person, so they can annotate it.
Yeah, and yeah. So it would be like,
Yeah.
and then there'd be like some sort of.
And we can potentially mention them. This could be a next project of DevoWorm. So if they wanna collaborate in the DevoWorm projects, they could just work with this at the starting point. So we can evaluate them who are interested in the project, who are not interested in them. Maybe if they can reach out by next piece of time, it could be helpful for you to filter out them who is like genuine and all this stuff.
Yeah, and of course you'd need to have a way to submit it to a centralized repository, I guess. I don't know if you could just put it on to use GitHub, and then you'd also need some way to like cross-check, because people
Actually,
may not.
I will create the README file for this.
Okay.
And create a repository, and I will host it on the drive for the dataset kind of thing, and I will provide the link. Who wants to download the dataset, they can just download it. I have the zip file over in my laptop, so yeah.
Yeah, and then of course there will be like a cross check, because unless, well, I guess you could just provide people with a template for like submitting information. Like for example, have like a format for the position or the name.
Actually, it is completely mentioned in the video, Bradley.
Oh,
I mean,
okay.
they can see the video, this tutorial, and they can annotate it as they
Okay.
mentioned in the video. That's it. We can cross-evaluate when needed. There.
All right. Yeah, I guess I don't understand that right now, but
No, yeah. That's These all should be like a pieces kind of thing. Once you watch this video, you will get a complete idea. What we can discuss is tomorrow after your watch.
okay. Yeah, that sounds good.
And Bradley, I don't have any update. I have mentioned you on Slack, I think so.
Yeah.
Maybe next week I will give you an update on the GSOC project stuff. I haven't worked with last week and before last week due to this all concussions. So yeah.
Yeah, that's fine. Yeah, so this looks good. This looks good. I'll
But
look at
before this, watch this video. If you have any other model organisms in your mind to fine tune it as a project, that would be great also. Because this works with any model organism, not only C. elegans. But I thought C. elegans because I know about it. Other model organisms, I don't know. We can give the cell names for each cell according to the position. Yeah, that's what I was thinking. If you have anything in mind, you can work with this kind of stuff. We can definitely work it.
Yeah, we do have axolotl data that Susan has provided, and those are open. I mean, they're a little bit different C. elegans. There isn't a nomenclature that you can attach. Like C. elegans, there's like every cell has a nomenclature identity, and and then but with the axolotl embryos, I guess they're a little bit different. But they can still, you can annotate the cells cell by cell, so it's not like it's impossible, or there might be some value in doing that. So I'll think about it in the context of how the software works.
Okay.
Yeah, that's great. Thank you, Sushmanth.
I was just wondering if Dick, could you upload the images from the recent axolotl batch that I imaged with the ball microscope? I tried to do that and it didn't work. I still don't know why. Is he there?
Is this
anyway? I gave a disk to him physically, USB disk to him with the images, so he does have them. It's just, but I did try to upload them and it didn't work. So I don't know what's going on with sync. So.
Right. Okay.
But I do have some images that would work. They're distorted as an oval in some instances, and they have shadows from the microscopes on some of the images. So you're working with things like that, but at least I have some. And I thought of maybe putting some more light around the microscope to get rid of the shadows, but I need to find some more salamanders willing to lay eggs.
Yeah.
Okay, Susan, I've had two problems lately. They upgraded Sync, and the new version didn't work on my computer.
Oh, fine.
And it took a couple of weeks to fix that. So I'm back to the old version. And the second is that I've exceeded the 2 terabytes of my internal disk.
Oh, okay.
Okay, so I still have your sticks, but that's it.
Okay, all right. There's Anyway, there are images.
Yep.
Just a lot of data, which makes them hard to handle.
Yeah. Well, yeah, I mean, I have some data that you sent me, and I have that in a separate repository. Maybe we could just use that. That would be like a good first pass, because I don't know. We'd have to work out a methodology. I also been talking to Hari Krishna, and he was mentioning that he wanted to have like I don't know, he was talking about a reference frame for the images. And I said that, that I don't know what he was referring to, but it hadn't been worked out. So he was going to try an automated method of that. And I'm not sure what he's referring to, but I guess I'm not really sure what he means by that. But I just didn't think that, that was something we had. So you might be able to provide something like that.
So, Bradley, should I send you a stick with the images on it?
Well, I have, you could do that. I have some. You sent me a stick a while back.
I still haven't sent you the synchrotron images.
Oh, okay. Yeah, I don't have those
either. So I'm sort of annoyed at myself for not getting there. But my computer upstairs works now because they installed fiber optic and a proper system, so I can now get onto the internet there.
Right.
And that, I don't know, that helps. Yeah. That helps the computer operate correctly. Anyway, so my upstairs computer is now working, so more access to images.
Yeah. Yeah. So that's
too many computer.
Yeah.
And I need a new one.
We can work on the axolotl maybe as well. So that would be good. We'll set that up. I'll take a look and we'll talk about that offline. So and then the idea would be we'd have. Let me see if I can pull up Hacktoberfest from, I guess maybe 2 years ago. I'm not really sure. So this is the DevoWorm organization. DevoWorm the repository, and we were doing Hacktoberfest in this repository. We have some. Well, okay, we have. So what you basically do for Hacktoberfest is you have the repository. Usually put a badge on it. I think we have the badge here. I don't think we have it right now, but we had a badge here. You can make a badge and put it on. And the idea would be you'd advertise this in places where people are likely to, be interested. So it would be like on social media or in the, you might talk to some people at a university or something. And as long as they know where it is, they can come here. They can look at the repository, look at the README. There's a badge usually saying that you're participating in Hacktoberfest, because people sometimes will come from, sometimes they'll peruse GitHub to look for open Hacktoberfest repositories. And so once that happens, they, can go in and look it over. And then one way to sort of facilitate a lot of these things is to set up a set of issues specific to Hacktoberfest. So I'm not sure if we have any that are from previous years. Okay, here we go. So when we did this, this was actually in 2020, these examples, but we had a couple of issues here. We have this label Hacktoberfest. So this is where you put this label on the issues, and it tells people this is what we want to achieve during Hacktoberfest. So we create the set of issues, and we give it a description, and then we give them instructions on what they can do. So here we had Mayuk set up an issue here, and then Ravi said, I would like to do this. And then Mayuk says, we're looking forward to a pull request. Another person who's interested in contributing to this issue. Ravi then issued a pull request that got merged successfully. And then I guess this is a different person. Yeah, this is a different person. Thank you for your interest. This is the second person. I would like you to look at contributing MD, which is a file in the repository, and check the norms used for a proper commit message. So you have to coordinate people's pull requests and all that, but basically that's how it works. And then if someone commits, like I think the general convention is if they make 5 commits during the month, they can get I think they can get a free T-shirt from GitHub itself. But usually we offer things, and like I said, I don't know what kind of position we're in to offer things. But I know that recognition is one thing, but that's less important right now. The important thing is that we get the details together. We can just tell people what to do and how to do it, and then we can even create a new directory for that. So in the repository, so in DevoLearn in the organization, we have the DevoLearn repository. We have other repositories like data science demos, DevoGraph. We can just create a new repository here. That's basically I don't want to call it Hacktoberfest. I just want to call it maybe whatever I don't know. We'll have to find a suitable name, but basically,
yeah, a datasets library. Right now we don't have the specified datasets to work with, so we'll create a repository called datasets, and whatever I have the dataset, I will upload over here. People can download it and annotate it. We will create issues in the GitHub page, 1 to 50 images, one issue, 50 to 200. For some people. Like this, people can just go and visit it and assign themselves that issue and work on that. Right now, I have another thing in my mind and issue in my mind. I will file it over DevoWorm and I will take permission from my. Is that okay, Bradley?
Yeah, that's good. Yeah, that's good.
Yeah.
So we'll create some issues, sort of, to start with, and then people can also assign. Well, they can assign themselves to issues. They, of course, can create their own issues, but yeah, the way it usually works is you start with a set of issues. Yeah.
Okay, I will create the datasets repository over DevoLearn, and I will try to create a structured one, and after approval from Mayuk and you, we'll make it as public.
Yeah.
And people can just come and just take up the issues, send to them, and work on it.
Yeah, that would be great. I have. So, and we have materials. I have to go back and look at it, but we have some materials for promoting Hacktoberfest. So we have a badge that we use for the org, and we have other things that we can. And then we want to get the word out, and so that's good. We also want to use the Hacktoberfest label for the issues because we want people to be able to find them on GitHub. And I don't know if, the Hacktoberfest label is in the DevoWorm repository. You may have to recreate it for this new one, but that's trivial. All right.
Yeah. Okay. We will discuss it further in this coming week, and we'll try to complete it by October 1st, so everyone can work on it.
Yeah, it'd be good. Yeah. Yeah, so in past years, we've had quite a few people contribute. We've had I think one year we had 11 or 12 people. So that's good.
Okay.
A nice pipeline into the org. And then, of course, this is something that people will use for, we can actually advertise, in fact, and say that this will be part of next year's GSOC. So people have extra incentive.
GSOC,
yeah.
People will show extra interest over this then. That would be, let's see what we can do.
Yeah, that'd be great. So yeah, that's great. So we're getting somewhat close to the end of GSOC the extended period. So both Sushmanth and Himanshu have been extended to the 24th of October. So coming up within about a month is the final submission for GSOC. And a few words on that. So, when you're working on a project, oftentimes you have to pick a point at which you publish where you submit some version of something. And so you don't,
yeah. We are working on it. Actually, I was working on the paper. Most of the coding part has been done. I need to restructure it and make a documentation for it. That's what was left right now. I will work this couple of weeks, maybe it will be completed. I'm just not concentrated last couple of weeks, 2 weeks. Next 4 weeks we have, right? Exactly. It will be completed.
Okay.
Yeah.
So I guess my point there was that at some point, we always make like the version that we release or we submit. And so we have, I guess, the way that Google wants to do this is to have like a submission, like a README or something that they can read through and they can look at the submission and be able to, actually download or run the software in some way. Now, the program has expanded a little bit this year to include things that are not software related necessarily, but just basically to have a place that's going to be a permanent home for the submission that has like a README that's very clear to read and so forth. So that's I can provide more details on that if you need, but just to let Sushmanth and Himanshu make them aware of that.
Yeah, definitely provide to us. We will go through it.
Okay.
I'll go through it.
Yeah, no problem. Okay, so why don't we switch gears here and talk a little bit about some things. I actually like to talk about some things on cell types and estimating cell type number and some new work that's come out on that. So I'm going to share my screen. And so there have been a number, actually, I think 2 papers that have come out, or actually 1 paper that's come out, and then some other things in support of that have kind of rethought this idea of counting cells and cell types in an organism. And so we've talked about this in past meetings. John Bonner, I think, published a book about 35 years ago where he was trying to make estimates of cell number and cell type in the human body. And more recently, people have made estimates of cell number and cell type, but they've been wildly variable. And one of the reasons is because what's the criterion that's used? What is the criterion people are using to determine what the cell type is? And that's no easy thing to sort of make a criterion for us, because there are all sorts of ways you can classify a cell. You can use phenotype, you can use function, and more recently people have started to use molecular markers. But molecular markers are pretty broad in terms of what to include, what to exclude. Sometimes you have markers that most cells have. Sometimes you have markers that are restricted to certain cell types. But it's hard to really kind of support that with other types of features. In other words, you might have a marker that's very specific to a certain sort of precursor cell, but the morphologies of those cells might be divergent. So it's very hard to do. And so there's this paper that came out recently in PNAS, and this paper is called The Human Cell Count and Size Distribution. So the authors are listed here. Looks like they're people from Stanford and other groups. And so they've done this in 2023 version of this. So I'll read from the significance statement, then I'll read from the abstract. So the significance statement here says a consistent and comprehensive quantitative framework of the cells in the human body. And so that's what we're trying to do. We're trying to build a quantitative framework, not just cell counts or cell type counts, although we can do that as well. And that's useful for the reasons why we just talked about doing machine learning and deep learning on microscopy images. And to do that, you need to have not only that segmentation worked out, but also sort of differentiating between types of cells in the images. So that's where it's useful. But having a larger quantitative framework, it could benefit many areas of biology. We compiled data to estimate cell mass, size range, in cell count from some 1,200 cell groups, from the smallest red blood cells to the largest muscle fibers. So these are all sorts of different cell types, cell morphologies, gene expression profiles, etc. This ranges across 60 tissues in a representative male, representative female, and 10-year-old child, and the gender isn't specified there. We find large-scale patterns revealing that both cellular biomass in any given logarithmic cell size class and the coefficient of cell size variation are both approximately independent of cell size. So we're talking about like a logarithmic transform and a coefficient of cell size variation. Those are the two measures they're using. And those are independent of cell size. So you can have actually information about the class and a measure of variation that are independent of the size itself. So as the cell gets bigger, these things are independent. These patterns are suggestive of a whole organism trade-off between cell size and count, which means that you have the count number of cells, the size of the cells. Those things, there's a trade-off between those, and imply the existence of cell size homeostasis across cell types. So this is where we talk about, how the cell is regulating itself, maybe its identity, maybe its size, just overall, and how that's regulated across different cell types. So if you have a certain role as a cell, you have a certain identity, that size will limit, it'll limit what size you can be, what the optimal size is for that function, etc. So this is the abstract. Cell size and cell count are adaptively regulated and intimately linked to growth and function. Yet, despite their widespread relevance, the relation between cell size and count has never been formally examined over the whole human body. And what they mean by that is that there hasn't been like a high throughput data set generated. People have made estimates, but there hasn't been this. I guess what they're doing is they're doing like a high throughput analysis of this. So here we compile a comprehensive data set of cell size and count over all major cell types, with data drawn from over 1,500 published sources. So this is sort of almost like a meta-analysis. And so this involves a huge number of sources of data. We consider the body of a representative male 70 kilograms, which allows for further estimates of a female 60 kilograms, and a 10-year-old child 32 kilograms. We build a hierarchical interface for the cellular organization of the body, and they have this app here, the Human Cell Tree Map, giving easy access to data, methods, and sources. In total, we estimate total body counts of the approximately 36 trillion cells in the male, approximately 28 trillion cells in the female, and approximately 17 trillion cells in the child. And these are, there could be different cell types in that count, but that's their estimate. These data reveal a surprising inverse relation between cell size and count, implying a trade-off between these variables. So basically the larger your cell size, the smaller the number of cells, and vice versa. Which makes sense if you think about if you think of the body as like this trophic system where you have cell types with these functions and trophic functions, and they have this sort of relationship between their size and their function and their proliferation ability, or the need to proliferate. So if you're a blood cell, red blood cell, white blood cell, there are many more of you than, say, if you're like a cell in the body that, like a neuron or something. And so these data reveal a surprising inverse relation, implying a trade-off between these variables, such that all cells within a given logarithmic size class, so they transform this into logarithmic size classes, contribute to an equal fraction of the body's total cellular biomass. So that means that you have these fractions of biomass, and that the size classes are roughly equivalent. So the smaller cells are more of them, but they contribute in proportional numbers. We also find that the coefficient of variation is approximately independent of mean cell size, implying the existence of cell size regulation across cell types. So there's this cell size regulation. You get to a certain size, you stop growing, and there's this size limit, And so again, this may be energetic. This may be something to do with function. This may just be something to do with kind of what's sort of the number of cells that are already in the body. These sorts of things. Our data serve to establish a holistic quantitative framework for the cells of the human body and highlight large-scale patterns in cell biology. Let me see, there was a comment in the chat. Dick says the size-number trade-off is similar to that of polyploid axolotls. The size-number trade-off, yeah. So yeah, polyploid axolotls have the same sort of relationship.
How many cell types are there? Did they actually come out and say that?
Let's see. They say 1,200 cell groups. I guess that's what their types are. So yeah, they don't. Let's see if they give a number. I mean, these are, I think, largely estimates. I mean, they do the counts, but you can't observe all cells necessarily. They're just getting the cells that they have access to. So here they talk about cell types. At one level, it is apparent that the body's cells are the right size to best perform their function. Irregularities in cell size are often a pathological sign of disease or a marker of malignancy. This means that, cells can go down a cancer pathway and start to, show all sorts of aberrant phenotypes. And so, but they're, technically that origin is from like one cell type or another cell type. So this is where, we kind of want to filter those out, but we also have variation, especially in cells like fibroblasts or something like that, where cell shape variation is where size variation can happen. Moreover, changes in cell sizes are associated with changes in biosynthetic capacity and metabolic function, especially when cell size changes are not accompanied by changes in ploidy, which is the number of chromosomes in the cell. Cell types such as myocytes, neurons, and adipocytes, which are fat cells, myocytes are muscle cells, vary extensively in size, but each cell has a size specificity suited to its function. So, you have these types that vary in size sometimes within the category, but each cell has a size specificity that's suited to its function. So, for example, muscle cells and sensory cells are smaller in the face than in the legs. So you have muscles in the face that are very small. There are a lot of muscles in the face, in the human face. And in the legs, you have larger muscles that span like the entire limb or the entire one segment of the limb. So, these are there's a relationship between sort of the nerves that innervate, like say the face versus the parts of the leg, and maybe the muscle size. And so we know that from developmental neurobiology. So, there are these relationships with the nervous system, relationships with the morphology, and the need to have muscles or, sensory fidelity in certain parts of the body over others. So this size homeostasis is thought to be maintained to some degree across all cell types. But the strength of size regulation, which is often measured as the coefficient of variation in size, remains unknown across the full cell size range. So at a higher level of organization, the overall functional capacity of a tissue depends on its total biomass. So this is, again, the tissue, if it's biomass, if it's larger, its function is different than if it's smaller, or what it can do. Since the cell types of any given type are relatively well constrained, the tissue's functional capacity is determined principally by the total number of its cells. So the total number of its cells determines its size. The size determines functional capacity. And so that's how they're sort of characterizing that. Two important studies in the past decade have surveyed the total number of cells over the largest tissues in the human body, with total counts converging on values of 30 to 37 trillion human cells. That's 16 and 17, which I'll get to in a minute, with an approximately equal number of bacterial cells. So cell size and number are direct corollaries of growth and proliferation, and among the most fundamental quantitative attributes of life's basic units, the relationship between cell size and number have never formally been examined with the whole body. So they do this sort of, they compile these size, abundance, and diversity statistics. Let's see, 16 and 17, these other studies that they're citing. So we'll get to those figures in a minute that I just kind of glossed past. But 16 is Bianconi et al., an estimation of the number of cells in the human body. And this is from 2013. So this is from Annals of Human Biology in 2013. So that is one paper that's attempted this in the, maybe within the last 10 years. Put that in the chat. And then we have 17, which is Sender, Fuchs, and Milo, revised estimates for the number of human and bacterial cells in the body. This is where we're looking at human cell bacterial symbiosis, but also the number of bacterial cells and living in relation to the human cells. So this is PLOS Biology in 2016. So there are a lot of really interesting citations in this paper, actually, now that I look at it. So I'll post this, or I'll send this paper out by email and post it in the Slack, and you can look at it more, at the reference list more, which might be very enlightening in and of itself. But let's go up to this. So this is let's see. So this is an image of different cell types. So we have platelet cells, which are very small, endothelial cells, stem cells, which are, a little bit bigger. Fibroblasts, of course, which have this morphology that branches out, but isn't like a neuron with long processes. It just kind of has these, looks like kind of a star or something like that. And, they can have variable shapes. Erythrocytes, neutrophils, alveolar, these are in the lung, alveolar, hepatocytes, neurons, of course. This is one example of a neuron. There are many different examples of neurons again. Very complex processes. Adipocytes, which are fat cells, and then myocytes, which are muscle cells, which are syncytial cells. So there are all sorts of cells that we have in the body, different sizes, different tissues, and they play different roles in the body. And so, this is where we get this above, we get these different cell types color-coded onto these bins that they talked about. So these are the bins where you're taking cell mass as negative powers of 10. So you get like negative grams, so 10 to the negative 4th grams, 10 to the negative 6 grams, 10 to the negative 8th grams, 10 to the negative 10 grams. So they're very small and they don't weigh very much in and of themselves, but in the aggregate, they're putting together these bins to show that the cell count goes up as the cells get smaller. So you can look at the size in terms of mass instead of looking at in terms of just measuring the size by looking at an outline and looking at the inside. We're just looking at mass, and then we're looking at cell count. The cell count goes up with reductions in mass, and this is roughly, I guess, logarithmic, and this is the equation here. So this is actually another really interesting table. This is where cell class distributions are sort of co-located with selective tissues. So we have all these different types of cells, lymphoid, non-nucleated blood, granulocyte, other blood cells. We have myocytes, cardiomyocytes, striated myocytes. We have endothelial cells, epithelial cells, stem cells, parasites, germ cells, neurons, fibroblasts, glial cells, osteoids, and adipocytes. And they all have these different color codes. And so up here what they did is they put this into different tissue types, or at least they collect them for different tissue types. So you have the cell count, which is an estimate, but it's basically a really large number in each tissue, and then you have cell biomass in grams, and then you have these different sort of fractions of each for each tissue. So for the testes, for example, you have this light green, which dominates the count, but maybe is a little less prevalent in the biomass. The light green being germ cells, of course. And then you have a darker green band, which are stem cells, and then you have other. So you can see that in each tissue, you have these different cell types. They have different roles to play. You also have the central nervous system, peripheral nervous system, adrenal glands, liver, intestines, and so forth. So you can see the cell type variation across those. In the intestines, you have a lot of blood cells. In the skin, you don't have as many blood cells. You have a lot of epithelial cells and endothelial cells. You don't have as many endothelial cells. But in the eyes, for example, you have more neurons. So we can go across the different cell type or the different tissues and look at the distributions that way. Then there's this other figure here, which is this figure 1. This again has these different cell types. So they give little pictures of them and they give like a color coding. And so this is actually a visualization where they put things in a sphere and they like take a fraction of the sphere and they have like a percentage. So it's like a way to represent different categories within categories. So for example, you have all blood cells here in the red, and within all blood cells you have white blood cells, nucleated red blood cells, and then within those you have lymphocyte T cells, band neutrophils, lymphocyte B cells, so you have these categories within categories. You have fibroblasts, you have stem cells, endothelial and epithelial cells. So you see that there's this, for the cell count, you have these different types of cells. You have a preponderance of white blood cells, and that's in terms of cell counts. So that's different from biomass, which is in B. So cell counts is in A, biomass is in B. Biomass, of course, by weight instead of by count, adipocytes dominate, or fat cells. And so you can see that, you have a lot of fatty tissue in your body in terms of biomass, but that's not represented by proliferation of cells. The number of cells that are adipocytes is rather small, but it contributes to a large amount of the biomass. Conversely, your white blood cells, the cell counts are dominated by white blood cells. They're very small cells that predominate. They proliferate a lot, but they don't contribute that much to the biomass. So it's a trade-off there, where you have weight of the cells, or what those cells contribute to tissues, into the body, versus the number of cells. So there are a lot of reasons why you'd have a lot of cells. White blood cells function as sometimes they need to be replaced. They need to be all over the body. There are a lot of reasons why you need a lot of white blood cells, but at the same time, they don't need to be very large to do their job. So when you have a predominance of those kind of cells in the body relative to other types of cells, they actually don't weigh that much. So that's what this graph is all about. Let me look at this link here in the abstract. So this is the human cell tree map. This is the tool that they have put together for this. So this is an app that you can open up on your desktop. This is that figure, and it kind of shows this distribution. And actually, you can explore it in this case. So we have the human cell tree map. So this is a tree map representation. And basically, it's like a tree that they visualize the different categories by size. So it breaks things down hierarchically into categories and subcategories. And then you can explore the different cell types here. So in granulocytes, for example, there are a number of different cell types that you can explore. This is both by count and by biomass. And so you have this graph here, the count on the right, you have cell mass, which is in grams, you have cell count, which is positive powers of 10. In cell mass, it's fractions of 1, so it's like you get 1e negative 4, 1e negative 11, that's the range. So they're in grams, so they're pretty light individually, but and then the cell count is, up to power 10 to the 13th. So we have we can put together, and again, these are estimates, but we can put together different counts for different collections of cell types, and we can get estimates that way. So they have that all plotted out for count, and then for biomass, this is done cell mass versus cell biomass in grams. So you're just looking at the mass of cells versus the biomass, which I assume is the tissue or the organism. I guess the tissue. So you have platelets, erythrocytes, so you have all these different cell types of different weights, and then the cell mass. So this goes through some of the different organ systems. So this is basically the tool, and this would be nice to play with. Again, the link for that is in the paper abstract. All right, so I have a couple of figures here. This is just a close-up of that figure 1, so this shows this in A and B. Of course, we went over this. The second figure is, and this figure 3, I think, that we looked at, where they have this logarithmic regression between cell count and cell mass and the different cell types. Yeah, this is the cell counts in humans. I wanted to go over that paper and some of the figures, because I think it's especially what we've talked about in the meeting, in previous meetings. We've talked about this topic before, and it's kind of like we've gone through the literature, and it's been very hand wavy in terms of the types and numbers of cells, and there's a good reason for that. But this I think is it's from what I can tell, a meta-analysis where they look at some of the features of the cells. I don't think they actually use any next-gen sequencing in this, but at least they don't claim to use the markers as any sort of identity. So they just go through the literature and look at what people have kind of come up with. But that's what we have, the state of the art in that area, I guess. Any comments or questions about that?
I think that my modeling of epithelial tissue only would be a good idea. There's too many cell types.
Well, yeah, in modeling, that's an interesting question. Like, what do we want to focus on in terms of cell type proliferation? Because there are different specialties, different organisms have different specializations. So this is just in human. So like Dick mentioned that, in axolotl, you can draw parallels, but like we don't have good, maybe in C. elegans we can do this kind of exercise where we look at because the cell types, of course, they're eutelic organisms, so they have the same number of cell types in every individual. So you have this in the lineage tree, of course, is deterministic. So we know kind of where we're going to end up. We know what the tissues are, and we could actually build that distribution pretty quickly for C. elegans. But for other organisms like axolotl, it's going to be different. You're going to have different types of tissues. The tissues are going to be composed of different cell types, and that's largely based on function. So where you have functional requirements that differ, you'll have different distributions of cell types. But like in terms of modeling, do we need to model every potential cell type? I don't know. I don't necessarily think we do. Although, modeling is an exercise in like parameterization, right? Like you figure out the range of values you need, and you don't know exactly what the cell state is. That's not maybe that's not the point of your exercise. The exercise is to see how basically a cell like that will behave under certain conditions. So, I have to stress that, like, cell types are very fluid in a lot of ways. Like, there's a lot of variation around the edges of those categories. They're not necessarily nice, neat categories. Sometimes they are, but oftentimes they're not, especially like across development or like, across people. You can have cells in various, states of life. Like sometimes they die, sometimes they're apoptosing, sometimes they're going down the cancer route, sometimes they're, interacting with other cells, and so
you'll
forth.
get epithelial to mesenchyme transition fairly simply by creating a wound or I don't know, stretching a tissue too much. If they lose contact with their neighbors, they tend to become a moving cell type, which is kind of intriguing.
Yeah, there's a lot of work on like wound healing that people have done, like both in the biophysics and in the identifying which cells are involved in which processes, because that's a pretty complex process where they have to migrate into the wound area and form a matrix that allows the wound to be closed, and then you have morphogenesis at that site. So it's a pretty interesting area.
Yeah, it is.
Yeah. So, do we have any comments or questions before we go? Okay. Like I said, we'll be working on Hacktoberfest for those interested. That'll probably be up and running by the first of the month, at least we hope. So I'll give people a whole month to contribute, and then we'll provide the instructions on how to contribute, and we'll probably do some getting the word out on that as well. So we'll probably be talking a little bit about it in the meetings in October. So look forward to that. All right. Well, thank you for attending. Have a good week.
Yeah, bye.
Bye. Okay, now I'd like to talk about 2 additional papers. And so these are on complexity and morphogenesis and phenotypic evolution. This is kind of a broad category, but you'll see where we're going with this. So the first paper is it's a scientific reports paper from this year, and the title is Robust Morphogenesis by Chaotic Dynamics. So it combines morphogenesis and some of the models we use for morphogenesis, and looking at the dynamical systems aspect of that. So a lot of the papers that we've seen talk about sort of different stating morphogenesis in terms of differential equations or in terms of cellular automata, and then showing that they produce these dynamics that are periodic. So we have things like Rule 20 or Rule 30 in cellular automata, where we have striping in our implementation of differential equations. So we have striping that's caused by different gradients, morphogenetic gradients. And so what they're saying here is that you can produce robust morphogenetic patterns or morphogenesis using chaotic dynamics. So the abstract reads, this research illustrates that complex dynamics of gene products, which are, of course, the expression of genes, and they're going to show that, this idea, the concept of a morphogen, which is basically just a generic vehicle for different types of gene expression and the production of gene expression, enable the creation of any prescribed cellular differentiation pattern. So this means that you can take this basic model of coordinated gene expression and you can produce different patterns that are very robust. They describe a lot of different modes of activity. They describe a lot of different regions of that space. These complex dynamics can take the form of chaotic, stochastic, or noisy chaotic dynamics. So they're in chaos and in dynamical systems. We have different regimes for chaos. We have different regimes for things that are on the edge of chaos. And we have different regimes for noisy behaviors. So they'll talk about these different types of dynamics. Based on this outcome in previous research, it is established that a generic open chemical reactor can generate an exceptionally large number of different cellular patterns. The mechanism of pattern generation is robust under perturbation and is based on a combination of Turing machines, Turing instability, and Wolpert gradients. Wolpert, of course, is Louis Wolpert, and this is the idea of morphogenetic gradients. Turing instabilities are chemical structures that exhibit instabilities at different critical points. And Turing machines are, of course, the idea of running a tape and, seeing if it halts at a certain point. So this kind of integrates a lot of different things. I've not heard of Turing machines really being used in morphogenesis. I've heard of Turing instabilities and gradients, of course, but not bringing that together with Turing machines. In any case, we'll see what they're getting at here. These results can help us explain the formidable adaptive capacities of biochemical systems. So, there's this idea that morphogenesis is self-organized. And it's generated not by like a centralized top-down mechanism, but by a set of bottom-up processes. So, for example, you can have regional differences, you can have differences between cells, between parts of tissues, even within cells. And I think in the last 2 years, we've seen a lot of examples of this in our readings in the group. So we've had different types of single-cell morphogenesis, tissue morphogenesis, organismal morphogenesis. And we've seen all these examples of that space is very important, spatial differentiation is very important. And you end up with these sort of gradients. You see things like striping or spotting or different types of bands that are formed in different patterns. So you get pattern formation, you get regularities. How do those things emerge? And the idea is that you have no central controller. You have these cells or these gene circuits that work in a bottom-up fashion. They coordinate themselves through different constraints, and they produce these patterns. Now, I might be tempted to say, well, we want to have a top-down mechanism, that we should have a supervisor that coordinates all of these spatial inhomogeneities and mechanistic inhomogeneities, but that's not actually the way it works. It does work through a bottom-up process. And so this is a very good example of emergence. And they're going to talk about, they're going to talk about the field in there and talk about this dynamical systems model. So, one idea is the idea of Louis Wolpert's positional information. And so positional information is where you have a cell, and it's in a location in an organism, where it's in a location relative to other cells. And that positional information can be used as a differential mechanism to describe, the formation of patterns. So why don't we draw this out? So positional information is this idea of where you have a bunch of cells. And why don't we just do this? Why don't we just have like a random cluster of cells so I can show you what the positional information is here. So each of these cells have a position. And let's say it's in an XY plane. And so we can describe each of these cells in an XY plane. And so, there's a difference here between Y. There's a difference here between X. There's a difference here between X. And there's a difference here between Y. So you have like these you can take differential measures of any two cells, a pairwise relationship, where sometimes you can take this in groups. You have clusters of cells that share kind of a common set of positions versus others, and so forth. So this is just a random collection of cells. Now, as we know, organisms are not random collections of cells often. Oftentimes they're regionalized so that you get very early in very early in embryogenesis, you get polarity. So you get EP polarity. Sometimes you get left-right polarity. And so we get this, this these different parts of the embryo. So the cells in these different parts of the embryo are going to be sort of have a positional identity. And so some of these cells might be in the anterior pole, some of these cells might be in the posterior pole, some of these cells might be specialized for the right-hand side and the left-hand side. Oftentimes, you'll have stereo pairs of cells. So you have, that's something that we see a lot in C. elegans, for example, where they have a common identity and then those identities diverge over time as specialization becomes more implicit or explicit over development. Positional information then is just the sort of the location of that cell. So for example, we can take this embryo and we can draw stripes across it. And we can say each stripe is defined by a set of cells. And each non-striped area is defined by a set of cells. Okay, so right there the positional information is that they either exist or don't exist within these stripes. And again, this doesn't need to be imposed from the top down. This could just be an interval sort of thing. So in Drosophila, you have a whole system for striping and segmentation, a set of genes that encode for these kind of setting up these gradients. But if we have these gradients defined, then we can actually have each cell within the stripe or outside the stripe express different factors that can actually might sort of diffuse out of the cell over space, and then those signals, being oppositional, come into contact and form a gradient. So that gradient becomes like the boundary between those two chemical gradients emanating from each cell. And so they set up these boundaries and you get striping. And so all you need to do is specify within a cell what its general location is. And again, you don't need to specify it. It's specified by nature. But also, if you were building an artificial system, you might specify this at the level of the cells, not at the level of the whole embryo. The point here is that it's a product of gene regulatory networks within the cells. It's not a product that we impose from the top down. And we can characterize this in different ways. We can characterize this with using something like a cellular automata, which is a grid of cells that interact. So if we want to have a gradient, we can have a cell here that influences its neighboring cells. There'll be a cell here that influences neighboring cells. And you can see very easily where the cell grid there will be a boundary here, for example, between these two cells. Because their influence is kind of meeting at that point, and so they're suppressing each other, and you get a boundary there. Or you could have something like a set of differential equations. So, we might look at a set of differential equations as a way to characterize, these coupled relationships. So that's where we're going with this. Now, what they're doing is they're plugging in dynamical systems into this. So they talk about Turing as a reaction-diffusion mechanism, which I kind of showed, that you have diffusion of factors out of cells, that there are reactions at certain points, and those certain points of interaction form boundaries. In both conceptual models, positional information and reaction-diffusion, an organism is represented as a pattern consisting of different cells. So this is just where you have the cells in different parts of the organism that end up forming different patterns. Both Turing and Wolpert's approaches assume that morphogens, and these are these theoretical constructs which are these nondescript factors. They could be, a certain, like in Drosophila, we know the specific genes, but in other organisms, it doesn't really matter what the genes are, what the molecules are, as long as you have these, they have a certain action. Morphogens can change cell states. Modern research has shown that French flag and reaction-diffusion models do not capture all details of real biological patterning systems. But neither of these capture all details of real biological patterning systems. So there's some work to try to reconcile this, and of course people have talked about the shortcomings of these models. So we're not quite at a full level of understanding of all this, at least through the current models. In this paper, they're inspired by the ideas of Brenner and Jacob, Jacob's ideas about evolutionary tinkering. So this is references 5 and 6. We show that a combination of reaction diffusion and positional information mechanisms give a universal robust pattern mechanism. So you can combine these two to produce a universal robust pattern mechanism. Brenner emphasized the importance of Turing's ideas in the context of his famous machine. So this is where we're taking Turing morphogenesis and mapping it onto the Turing machine. It's interesting that Turing came up with these ideas but never really pulled them together during his life. And so this is, I think people are kind of thinking, well, pattern formation and morphogenesis is a kind of computation. And so they're making this connection. A Turing machine is a mathematical model of computation that defines an abstract machine that manipulates symbols on a tape according to some rules. The machine stops when it has arrived at a prescribed terminal state. In its turn, Jacob introduced the concept of tinkering. Evolution uses all available means. So this is where you're talking about this tape. It has different symbols on it. The machine stops, in this case, when it arrives at a prescribed terminal state. And so the idea is that you can tinker with that state. You can tinker with the tape. So the tape has all the states stored on it. And you just run the tape until you hit the right combination. We show that the dynamics of a generic open chemical reactor, so they're using an open chemical reactor, is capable to generate an exponentially large number of different attractor points, which may be chaotic, periodic, or of a steady state nature. So what they're doing is they're saying that these symbols, or these symbolic states on the tape, can be represented by attractor points in a dynamical system. The chaotic or periodic attractors can occur if our reaction diffusion model is not gradient-like and not monotone. So this actually speaks to this reaction diffusion model not being gradient-like. We also don't want it to be monotone. So this is kind of interesting because it means that you can have chaotic patterns form. So there's no linear gradient. It's not, kind of a smear. They're different, there are ways you can have like kind of local states that are differentiated. The results hold under certain mathematical conditions which admit a transparent chemical interpretation for two-component systems. In this case, one of the reagents is neither an activator nor an inhibitor for the other reagent. Reagent diffusion coefficients are sharply different, and the space dimension of the system is greater than or equal to 2. Okay, so that's the first result. So we have a two-component system. One of the reagents is neither an activator nor an inhibitor for the other reagent. So you have two reagents, they're kind of independent. The reagent diffusion coefficients are sharply different. So that means that the reagents are different in some way. And the space dimension of the system is greater than or equal to 2. So the space is larger than the number of reagents. The second result is that such open chemical systems can be used as sort of a Turing machine, which can produce an exponentially large number of different cellular differentiation programs. So this is where they define CDPs, or cellular differentiation programs. This is a program by which morphogen concentrations produce cell patterns unfolding in space and time. So for example, you can have gastrulation patterns, or somatogenesis, which generates periodical structures. And the number of possible cellular differentiation programs increases exponentially in the system size. So this is kind of, you can think of cellular differentiation programs maybe like an instruction set. But maybe that's not the right way to view them, but that's what they're going with here. The proposed cell pattern formation mechanism is robust against fluctuations and perturbations. So, a combination of two of Turing's basic ideas, machines and instability of chemical systems, leads to general results, which sharply extend Turing's instability model to a computational realm. The combined Turing-Wolpert model makes it possible to obtain evolving over time patterns that are much more complex than zebra stripes. So they give the example of zebra stripes. It's that striping example that we saw here. In this slide, I mean, you could see this on a zebra where you have black and white stripes. They give that example, but what they're saying is they can generate different types of morphogenesis and different patterns that are much more complex than zebra stripes. So they're not linear. The mechanism of the attractor and pattern generation can be sketched as followed. One can show that a generic open chemical system is capable to generate a number of chaotic or periodic attractors if it involves appropriate spatial heterogeneity. So these are these heterogeneities we talked about before, where things are different across space. You have to have, a great deal of spatial heterogeneity in order for this to work. These inhomogeneities work as activators and excite the formation of sharply localized segments like Drosophila segmentation, which we mentioned. And there's specific genes that are involved in Drosophila segmentation. Segments correspond to local maxima of slow reagent concentration. So this is where, I guess, the speed of the reagent concentration, or a different type of reagent concentration, you get these local maxima and you get segments there. This doesn't necessarily map to a biological system. This is just a model that they've developed. So it doesn't necessarily map to Drosophila segmentation, but does explain Drosophila segmentation, which is interesting if we know a mechanism is involved in something. And then we have another model that explains it just as well, that maybe isn't based in the biology so much, but can replicate it. Then it's kind of interesting because we don't know, maybe the solution that happened in nature is one possible solution to this. Maybe there are other ways to do that. So it's kind of an interesting lesson sort of in evolution. Segments correspond to local maxima of slow reagent concentration. These segments interact in a long-range manner via the fast-diffusing component. All these mechanisms reminds one described in the manuscript, but we are capable to prove that these lead to chaos. The chaotic dynamics allow us to generate complicated cell patterns in space and time. It can be shown by the results from these works. Okay. So maybe we'll go to some figures. Figure 1 here is this picture illustrates the concept of a developmental program. On the x-axis, which is here, there is a one-dimensional organism composed of various cells that form layered patterns at different time points. Different types of cells are shown in different colors, so the red versus the blue versus the green versus the black. The top row of cells emerges as the last moment, Tn. The previous row appears as T equals Tn minus 1, and the bottom row rises at the initial time moment. So this is just kind of describing this pattern. The x-axis is this one-dimensional organism. They form these layered patterns. These layered patterns are these cell types. These cell types, you can see that they're stratified across the organism, and then they arise at different times. So I'm assuming, and they don't label this, but the y-axis is time. So you get these emerging over time. So you basically get this layered structure that kind of emerges over time. The farther down the organism you go, the more tissues you get, and that unfolds over time. So you can see that morphogenesis proceeds apace. We refer to this two-dimensional spatiotemporal pattern consisting of cells of different discrete types as a cellular developmental program, CDP. So you can see that figure 2 shows the generalized French flag model. So the French flag is 3 colors, and you have to achieve these 3 color schemes as layers. So this is not just the two, the on and off striping of this example, but this is actually 3 different states kind of layered like so. And so this image shows a generalized French flag model. A cell pattern can be considered as a string of the alphabet, red, blue, or green, so it's this alphabet of size 3. This pattern can be produced by chaotic dynamics as follows. Let U be a vector of morphogenetic concentrations, which lie in a morphogen concentration space script U. Suppose that the space is split up into 3 subdomains, script U sub J, J 1, 2, 3. If U is included in this script U sub 1, it's one of these numbers, one of these in the index. Morphogen concentrations induce differentiation in a red cell. And if it's included in script U2 or script U3, then one has a blue and green cell respectively. So basically, you have these different, these different concentration spaces. And if they're included in one or the other, so you have this index 1, 2, 3, if U is included in one of these, then it's going to express a different color. And so when U dynamics is governed by a chaotic system, there exist moments in time such that U state enters for script Uj at a certain time. Moments, there exist T and then change in T such that the state changes lie in the corresponding domain and then this domain. So basically what you're getting is this this three-state system that's being generated over time. So you can see these are the same axes as in Figure 1. So this is on the x-axis is the one-dimensional organism. On the y-axis is time. So as time unfolds, you get a more complex organism, but you get this striping where you get state 1, the blue state, the green state, the red state, the green state, the green state, the blue state, the blue state. They're just generated in this way. So over time, you get this larger organism with more layers of different. You only get 3 tissue types, but the tissue types are variable, and they don't all draw from the same. They don't draw the same tissue type. Sometimes you get paired, like green, green, and blue, blue, and red, red. But it's a stochastic process as to what the layers are. So let's go into the discussion here. So they consider phenotype morphogenesis and its robustness. The pattern formation robustness problem, initiated by seminal papers 15 and 16, have been an intense discussion for the last few decades. A number of different approaches have been proposed. Heat shock proteins and other molecular chaperones, methylation, microRNAs, emergent or embedded mechanisms, which are based on gene regulatory networks, and other nonlinearities in development. These are all things that have been proposed for this. So what they're saying here is that we can model this using dynamical systems theory and bring this idea of computation into the sphere. So it's kind of interesting. The general idea can be formulated as follows: timing instead of tuning. We can take any dynamical system, which generates complicated Turing pattern dynamics or Turing complete dynamics, and we know a number of such systems. And then if a target pattern is predetermined, these dynamics generates the pattern. The mechanism of pattern formation is robust with respect to noise and other perturbations. And if it goes up for several stages, each of which, upon reaching a certain terminal state, ends up with a stop signal. So such multi-stage morphogenesis, where each stage finishes with a stop signal, is robust with respect to perturbations. The idea that evolution can use any means to produce effective structures, which is called evolutionary tinkering, was proposed by Jacob and Six, and we're going to go down to the references in a minute. Here we assert that practically any dynamical system with complicated large time behavior can be used to generate complicated patterns. Now that's an interesting assertion because we know that cellular automata can do this to an extent, and they don't mention that at all in this paper, by the way. This is just kind of going through Turing machines and linking that to Turing morphogenesis. But we know that cellular automata can do a similar thing where you can generate patterns given a sufficient set of rule set. And so, we see sort of how computation intersects here. The other thing is that I'm not quite sure that, I don't know. They didn't give a great demonstration of this. And, the stop signal is interesting, this idea of stopping the tape at the proper symbol. At the molecular level, of course, we have stop codons. And a lot of that is, very different from what we're talking about here. But this idea of being able to stop reading a gene at a certain point and producing an outcome, there's kind of a connection there. I don't know what that connection is, but it just kind of popped into my head. So note that the experimental data show that many genes exhibit an oscillatory behavior. So a lot of times gene expression is the output of gene regulatory networks is oscillatory. It could be over a day, it could be over a month, and patterns usually don't form instantaneously, they form over time. And so these genes can exhibit oscillatory behavior. sometimes a cell cycle, sometimes over longer time scales. And so we have to consider that as part of the circuit as well. Maybe the oscillatory behavior is being used to generate patterns. So, for example, if there's a pattern that's generated in space and it's also oscillatory with respect to time, that the spatial pattern will be discontinuous because the peaks of the oscillations are reached at different time points, and there's an interval in between where you don't have peak behavior, so you end up with these stripes. You may also have something like, where you can't form a pattern over time. Sometimes you can't form patterns, and then sometimes you can form patterns. So, for example, if a gene turns on maybe every night, or it's photoperiod dependent in some way, then you can have pattern formation during the day, but development proceeds all throughout the clock. So at night you won't have pattern formation. It's stuff like that. That's actually kind of interesting. A model describing the chaotic behavior of transcription factors is proposed in the research, where it is shown that such a chaotic behavior could be useful for survival and leads to the formation of heterogeneous cell populations. So they used oscillatory gene dynamics to describe somatogenesis, but that's something I think that needs to be thought about a bit more. I'm thinking primarily in terms of the outputs of gene regulatory networks, which we've talked about. These results show that there are chemical systems which would be considered in the light of analogy of universal Turing machines. So these are universal computational machines like cellular automata, but they're saying that these are universal Turing machines. A universal Turing machine can make all computations which can be done by other Turing machines, and so UTMs, or universal Turing machines, generate all possible string outputs, when we vary their input. In our case, we have fixed up to a few parameters to adjust spatially extended systems, which, depending on our initial data, generate different developmental patterns. So we have these spatially extended systems. They generate different developmental patterns. And so we're able to we have these universal generators of spatiotemporal patterns. And a few genes are sufficient to encode these. It's natural to expect that such chemical media could appear as a result of biological evolution. All right. So let's look at the reference list. There are a couple papers here. Sidney Brenner's Life's Code Script, which was published in 2012. That's one reference for this. And this is basically arguing that, I guess development is some sort of like code, or there's some code of development. This is interesting. We've talked about the differentiation code in this group. There's also positional information. But basically, there's this idea that there's some sort of plan. And maybe that's true, maybe that's not. But, it's worth thinking about. This is Jacob, Evolution and Tinkering from 1977. This is Francois Jacob, and this is another paper where they talk about tinkering and evolution, which means that you have a basic body plan that emerges, and then it's a matter of refining that body plan and shaping it in different ways through development. There are a lot of papers in here talk about Waddington, Siegel, and Bergman, which is Waddington's Canalization Revisited. That's an interesting paper. Levy and Siegel, Network Hubs Buffer Environmental Variation in Yeast. So this is a paper where they talk about network biology and environmental variation being sort of having a buffer, or the structure of gene expression networks being able to buffer environmental variation. And then this one here, Hava Siegelman, Computation Beyond the Turing Limit. This is in Science from 1995. So there are a lot of different, types of things that sort of they're drawing from here, from complexity theory, from biology. Okay, so the second paper I want to talk about is this paper from the bioRxiv, and it's also very recent. And this talks about Morangione-like tissue flows. So I don't know, we'll find out what those are. But these are, tissue migrations that are occurring in these flows. And this certain type of tissue flow enhances symmetry breaking in embryonic organoids. So lot of, they're all French authors. Last paper, I think, were a bunch of Russian authors. So, and so the abstract reads, during early development of multicellular animals, cells self-organize to set up the body axis, such as a primary head-to-tail axis, based on which the later the body plan is defined. Okay, so this is where the body axes are set up from these primary axes, and this is what defines the body plan. And they're saying that cells self-organize, so they don't have this top-down plan, which they're using. They're drawing from like local interactions in a spatially heterogeneous. Several signaling pathways are known to control body axis formation. And this, again, these gene regulatory networks that are controlling the formation of these axes. So it's axial control from individual cells, depending on their position in that mass. And then you get this sort of order, which is based on these axial cues and axial structure. Here we show, however, that tissue mechanics plays an important role during this process. So they're entering tissue mechanics into this. This is different from the last paper where they talked about Turing morphogenesis and reaction-diffusion, which is basically the diffusion of molecules across space, and also positional information, which is just position. We don't care about mechanics. So why don't we talk about this a little bit and draw this out? So again, we have our positional information. We have our spatial heterogeneity. So let's do this. Let's draw an embryo again. And we have our cells, and they have their own positional information, right? We have the spatial inhomogeneity. So a cell here is going to be different from a cell here. This is not positional information. This is just the nature of the cells. They're different in different ways. And so what do we label spatial inhomogeneity? I'm not going to denote that, we'll just keep that in mind. Then, of course, we have these local interactions, these reaction-diffusion interactions that occur between cells. And they set up a boundary if they kind of interact. Otherwise, they just kind of have this gradient around each cell. The gradients intersect at some point. And that's enough to form pattern formation. But what they're saying is that they're mechanical forces. So these cells just don't sit in the embryo just in one place and then form these patterns. A lot of times they move around this embryo in different patterns. So sometimes they move collectively, sometimes they move at random, but they're always migrating around from their origin point. So a cell divides, it produces two daughter cells. Those daughter cells will migrate. Now, as they migrate, they also produce forces. They're also subject to flows and forces of other cells moving, so other cells are moving around collectively. Sometimes they encounter jamming, which means that there's such a density of cells that they stop moving. So these are all, we're subject to all of this. So our pattern formation is complicated by motion and by this sort of, these patterns of motion. They didn't talk about this in the last paper, which is kind of interesting because, if you're talking about pattern formation as a dynamical system, this is probably something you should probably bring up. In any case, we have flows, we have migration, which are flows and jamming. And of course, interaction of forces. So these cells, as they're moving against one another or away from one another, or even as they're interacting chemically, they also have forces that interact. So if it's a matter of like cells squeezing in between other cells, or cells being blockaded from a certain place by a jam of cells, or something, that's interaction of forces. cells produce force as they move. Sometimes they are on different parts of the embryonic substrate, so they move differently. These sorts of things. So this is where they're coming from here. So tissue mechanics play an important role. We focus on the emergence of a primary axis in initially spherical aggregates of the mouse embryonic stem cell, or an aggregate of mouse embryonic stem cells in the mouse embryo. So the mouse embryo starts as a mass of aggregated spherical cells. This is an artificial system that they've made up. It mirrors events in the early mouse embryo. So they basically have this spherical aggregate of stem cells. This is something you see in the mouse embryo, but this is not the mouse embryo. So it's almost like in one of these embryoid models where you're trying to approximate certain things about early development. These aggregates break rotational symmetry to establish an axial organization with domains of different expression profiles. For example, the transcription factor T-bra and the adhesion molecule E-cadherin. So this is adhesion to a substrate or the expression of transcription factors that are very specific to axial organization. Combining quantitative microscopy and physical modeling, we identify large-scale tissue flows with a recirculation component and demonstrate that they significantly contribute to symmetry breaking. We show that the recirculating flows are explained by a difference in tissue surface tension across domains, akin to Marangoni flows. Well, Marangoni flows are a specific type of flow with specific type of dynamics. So we'll talk about what that is, which we further confirm by aggregate fusion experiments. Our work highlights that body axis formation is not only driven by biochemical processes, but that that can also be amplified by tissue flows. So this is where we have this pattern formation reaction-diffusion model. We have other types of things going on, and then we have these flows that actually amplify some of the pattern formation. And the pattern formation then also contributes to symmetry breaking, which we've talked about in some of our work, but if you need a refresher, or I'll kind of explain this to you a little bit. So you have an embryo which is sort of symmetrical here. It's exactly symmetrical. You have two poles, an anterior and a posterior pole. So this is the point at which they're symmetrical. But oftentimes, even when you get this pole start to be established, as in C. elegans, the posterior pole may be slightly smaller than the anterior pole. So you don't really have clear symmetry there. AP axis is asymmetrical at that point. And then as both of these poles, the cells start to divide and differentiate, you get further differentiation of the symmetry. So you get symmetry breaking events. And this is a term borrowed from physics, where people talk about symmetry breaking, where you have a symmetrical thing, and then suddenly there's an asymmetric shift. So that's what they call symmetry breaking. The symmetry is broken at some point. And usually you can think of this as a tree, so you can think of this as like a symmetry that's broken here. This is a pure symmetry up here. The symmetry is broken here. It's further broken here, and then it's further broken here. And the analogy in an embryo would be where if you had differentiation between an anterior and a posterior pole, and then that posterior pole got smaller, and maybe it differentiated into a germline, and maybe some tissues that were specific to the posterior end of the organism, and the anterior end maybe you formed a nervous system, some other structures. But what actually leads to more symmetry breaking is if some of these cells start to migrate collectively into different locations, which means that those cells are, not like contributing to this sort of equal mass. And so you get this differentiation, you get the symmetry breaking, leads to tissue formation, it leads to other things. So any one germ cell that you take, any one stem cell, as it divides, it differentiates, it forms a structure, it differentiates. So if you go to back to that mouse, massive mouse stem cells, you just have a bunch of individual cells that look basically the same. They do basically the same job. They're not in the same position in the mass because there's some spatial variation in terms of position, but there's really no variation in terms of signaling or in terms of physics. It's just this well-packed mass. Now as this massive cell starts to differentiate and the cells divide, you start to get things like this, where you get differences in position, differences in the location, and maybe like migration outward into little modules. They're like little arms coming out. And so you get a bunch of different types of variation here in terms of how these things differentiate. Things flow outward, where they flow, around the cell, outside of the cell mass. So the cell mass, because less of a spherical mass and more of sort of an irregular grouping, but you also get these clusters of cells that begin to have different functions. And so these aren't, uniform. You get chemical signaling between them. You have positional information that defines where they are. In this case, positional information on the left side, in A, positional information is less important because it's just kind of like, you're either, you're either in the middle of the pack or on the outside. There may be some differentiation there, but it's minimal. In this case, if you're on the outside, there's quite a difference between being on the inside and being on the outside. In B. Migrations complicate this because they move things around and introduce new spatial homogeneities. So what they're saying is this amplifies the existing processes, which is a perfectly fine statement to make, and it's probably true. But this is the kind of thing people use these terms that are very, they kind of come up with terms that sound good, but then what is the amplification mechanism? Can we measure how much it amplifies things? That's another story. Our work highlights that body axis formation is not only driven by biochemical processes, but it can also be amplified by tissue flows. We expect this to operate in a number of contexts. So we know that there are a small number of conserved morphogens, including Wnt, BMP, active and nodal, and FGF in animals that control the emergence of body axes. These signals participate in tissue patterning and trigger differential behaviors, which ultimately partition an isotropic group of cells into a different territory. So this is what I mean by the symmetry breaking, that these events actively participate in this progressive symmetry breaking, where things become more and more differentiated. They talk about tissue mechanics and flows being crucial for these stages of symmetry breaking. So it's not just genes, it's not just chemical signaling, it's not just its position, but it's migration, it's like mixing things. Ironically, this mixing contributes to symmetry breaking. And it's because it doesn't mix randomly, it mixes in a certain way that moves things to certain places. We know that tissue mechanics and flows can contribute to embryonic axis formation in avians, but we don't know the exact roles in mammals. So here's an example of what they're doing here. They look at these tissue collective flows. They have this this vector map here where they're showing the direction of the flow. Things are moving in basically in a coordinated direction amongst a bunch of cells. They're moving posteriorly a lot of the time. So this is contributing to symmetry breaking. You have this live imaging of this sort of migration, and then you have expression of brachyury, you get here, and so then also snail. This is live imaging, this is immunostaining. Just showing the different patterns of how things are expressed. So this is actually gastroloid symmetry breaking. So this is in a gastroloid. This is kind of showing this process averaged over 1 hour. They find that tissue flows substantially contribute to gastroloid symmetry breaking. We actually see that you have these different sort of stages of things. So you have reaction-diffusion, part of their model. So you have cell differentiation. And then you have diffusive motion. So there's this reaction-diffusion aspect where things move around, but it's diffusive. It's according to, say, like Brownian motion. They don't move like in a coordinated manner. They just kind of wobble around. they have this positional identity, but there's diffusion of things. And then you have this advection stage. So you have diffusive motion, which is basically Brownian, and then you have this collective motion, which is non-Brownian. It's super linear, and so the cells move around in a coordinated manner. This is what they call advection. So things are moving around. It's becoming well mixed, but because these cells are, have different identities, and they have maybe a memory of this identity, it's going to contribute to symmetry breaking ultimately. So you have collective motions, as we saw in Figure 1, and they move around together. And then you have growth, which is cell division and apoptosis. So some cells die, some cells proliferate, and that's how you get the progressive symmetry breaking. If you think about the cumulative change of polarization, which is something they measure quantitatively, and looking at hours after aggregation of these cells in this mass, remember the mouse cell mass, you see that you can look at different regimes here, and if you only consider one contribution of a single factor at a time, you get this graph here, 2E. So 2E shows that you have hours after aggregation from 76 to 92 hours. Cumulative change in polarization going up as the y-axis goes up. And you see that the total, it goes up over time. 76 hours is hardly visible, but at 92 hours becomes predominant, and actually after 80 hours. Reaction diffusion seems to be a major contributor to this. Advection is another contributor. Three-dimensional effects and growth are not really that much of an effect. Tissue flows also exhibit a dominating recirculating component. So that means that there's a lot of fluctuation, there's this recirculation of cells. To this end, we defined a disk that encompasses most of the aggregate projection and express the coarse-grained tissue velocity in polar form. So they put this in polar coordinates. We then decompose both spatial functions into different modes, again using polar coordinates, and they look at the circulation here. So this is analysis of tissue flows during polarization. These are coarse-grained tissue flows analyzed in a circular disk. And this shows kind of what they look like. So there's this recirculation mode where they kind of recirculate back. So you can see that they move in sort of the same pattern. These recirculating flows can be driven by pattern-induced tension. So in understanding the tension and how patterns form forces in the cell, so you have no pattern and then you have the onset of a pattern. And that pattern then has consequences on the embryo, on the cells. So cells there are induced tensions that can force cells to migrate after pattern formation. So this is not just a one-time thing. It's not just you form a pattern and that's it. So then they talk finally about the Marangoni effect. So we expect that Marangoni effect-driven recirculating tissue flows operate in many organoid systems. Especially systems formed from pluripotent cells. So in this case, we tested stem cells, but there are other types of pluripotent cells we can use. While such Marangoni flows can be artificially induced, and what they mean by Marangoni effect are these recirculating flows that we see up here. So you can see this very clearly here where you have experiments where you show this recirculating flow, and then simulation you can reproduce it. While such Marangoni flows can be artificially induced, they can also be driven internally. The emergence of such flows requires only the existence of distinct cell populations, which will typically display different surface tensions, thus providing a general mechanism for amplification of symmetry breaking. For instance, recirculating flows have been identified during mouse heart morphogenesis, or during the creation of the first cardiac crescent during gastrulation. So this forms a feedback loop that we can observe in potentially many systems. So that's all for today. Hope you learned something.